So let's move on to the reason we came together. I'm very honored to introduce Professor Alistair Boxall from the Environmental Science Department at the University of York. Professor Boxall is the theme leader of the Sustainable Environment theme of the York Environmental Sustainability Institute. Alistair's research focuses on understanding emerging and future ecological health risks posed by chemical contaminants in the natural environment. Alistair is a past member of the DEFRA Advisory Committee on Hazardous Substances and past chair of the Pharmaceutical Advisory Group of the Society of Environmental Toxicology and Chemistry. He regularly advises sorry, could you please mute yourself?
כל השנים היה לנו תוכנה בצד שנקראת לפידפו.
Yes, so sorry. Professor Boxall regularly advises national and international organizations on issues relating to chemical impacts on the environment and has published intensively on the detection, fate, effects, and risks of emerging contaminants, including pharmaceuticals, nanomaterials, and transformation products in the natural environment. Alistair leads the York City Environment Observatory Initiative and RCUK/Innovate UK funded initiatives to establish York as an exemplar city for understanding the linkages between the quality of city, environment and health, well-being and the economy of a city. So after this opening, I would like to give the stage to Professor Boxall, who will tell us today about the global problem of pharmaceutical pollution.
So thanks, Orly. I think I must update my website because most of what you said is very out of date. But hello everyone, and I guess first of all, sorry that we can't all be together in person. I much prefer giving these talks in person, but I guess that's one of the results of COVID. So what I'm going to do is spend about 45 minutes talking about the work that we've been doing, along with some of the other people in the room, actually, looking at the problem of pharmaceutical pollution in the environment. And before I get going, I'd just like to thank a few key people. So John is a postdoc in my group. Emily, Ali, and Andrea are either past or current PhD students in the group. And then finally, the group that I need to thank is the Global Pharmaceutical Monitoring Consortium. And a lot of what I'm going to present is actually a result of the cooperation with the members of that consortium. In total, we use as a society about 2,000 different active pharmaceutical ingredients. So if you go to a typical bathroom cabinet, you probably see something like this. And then you can see in the top shelf in the middle there, there's a box, and I think this is quite typical. And if you look closely at that box, it's packed full with different pharmaceuticals. So you've got creams, you've got tablets, sprays to treat a whole range of things. So hay fever, so I'm suffering from hay fever at the moment, so I apologize if I sneeze during the talk. But there's hay fever, and then things that the doctor will prescribe. And when we use these things, what will happen is we'll take them into our bodies, we might metabolize them, but ultimately what will happen is we'll excrete them. So we'll excrete the parent drug and also metabolites when we go to the toilet effectively. And where I live in the UK, and I think it's probably true where you live, what will happen is that will then enter the sewer network. So this photo here is actually one of the old London sewers. So this is a Victorian sewer. So this is what we still use in London. They are updating it, but this is the original sewer network of London. And you can see the sort of nice moody picture of the sewer. That water looks quite nice, but if I took a sample of that water, first it would be a bit smelly, but it would contain a real mixture of pharmaceuticals. So it will be the things that the population of London are using. Now that wastewater then will be transported down to a wastewater treatment works. In the UK, we use two types of wastewater treatment works typically. So this is the sort of works that we'll use in a more rural location. So this is what we call a trickling filter. And what you have here is effectively a bed of gravel. And on that gravel, you have a biofilm. And it's quite simple. You effectively spray the wastewater over that biofilm, and the population of that biofilm would degrade the organics in the wastewater. So coming out of the bottom of the filter then is fairly clean water. We also have a much more active process. So we have what's called an activated sludge treatment methodology that's used. There's different ways in which that's done. That's much more energy intensive. So in that system, you've got big vats of sludge bubbling away in water. So a lot of air going into it. And the bugs in that broth then are degrading the organics. And again, they're cleaning up the water. And that's what we'd use in a big city in the UK. Now, the problem with these plants is they were never, ever designed for pharmaceuticals. So while they're quite good at removing some pharmaceuticals, so something like paracetamol is actually quite well removed by these processes. For other pharmaceuticals, what you'll find is what comes in the wastewater comes out the other end in the water, and then it will be released into a river. So this is my local river. This is the River Ouse, which is the big river in York. So if you've never been to the UK, I highly recommend coming to York. It's a very historic city, very beautiful city, and we have a great river. But there are pharmaceuticals in that river. And we've, over the last few years, been exploring how the concentrations of pharmaceuticals vary spatially across the river systems of York, and also how they vary temporally. And we've also been using that data to begin to look at what the potential ecological risks are from exposure to these compounds. In terms of the monitoring, what we do is this is a schematic of our system. So you can see the bigger river to the left of the diagram. So this is the River Ouse, sorry. So each of those points labeled O are different sampling points along the river. We also have a second river. This is a slightly smaller river. This is called the River Foss. So you can see that along the foss, which is labeled F, again, we've got a number of sampling points. Now, York itself is the centre of York is effectively where the River Foss and the River Ouse meet. So what we've got is two rivers coming into the city, and then they combine, come out of the city, and then they flow down to the Humber Estuary. We have 3 wastewater treatment works in the city area. So on the Foss, we have a wastewater treatment works treating about 18,000 people's wastewater. On the Ouse, we have plant B, which is as the river comes into the city. That treats about 26,000 people. And then plant C, that's our biggest plant, that treats wastewater from about 175,000 people. So you can see that what we're doing with this design is we're able to look at the influence of the wastewater treatment works on levels of pharmaceuticals in the river system. But we're also able to sort of look temporally as well. So we take samples every month. We've done that probably now for about 4 years. And what we do with those samples then is we then take them back to the labs in our chemistry department. There we've got liquid chromatography with tandem mass spectrometry. And what we do is we analyze those samples for a real mixture of pharmaceuticals. So you can see there the types of things we look for. We look for things like analgesics, antibiotics, diabetes treatments, beta blockers, and so on. And we keep adding to that list, so that number keeps on growing. Now, I could spend an hour talking about the York data, but I'm not going to because that's quite local and really probably mainly of interest to people in York. But I'll just give you a very high level overview of what we find. So this is basically the high level data for 1 year. What we've done is we've calculated the average concentrations for each sampling point, and then we've plotted the median average concentration for the whole river, and also the maximum sampling average concentration. So on the x-axis there, you can see that we've got the different active ingredients that we monitor. So this is for the river ooze. So we monitor over 20 different pharmaceuticals. So at this stage, when we're doing the study, we typically look for about 40 active ingredients. And you can see that the highest concentration we see is for metformin. So metformin is a diabetes treatment. It's used to treat type 2 diabetes, and it's one of the biggest used drugs in the UK. And that's closely followed by gabapentin, which I think is used for nerve pain relief and also as an anti-epileptic. This is the river phos. Actually, before I go to that, I'll just go back. If you look at the y-axis, that gives you the concentration. So you can see in terms of metformin, the maximum concentration that we see is about 3,000 nanograms per litre, so 3 micrograms per litre in the river. This is what we find in the river Foss. So the river Foss, you can see there's a picture in the middle there. It's a smaller river. Again, we find metformin and gabapentin at the highest concentrations. We detect more pharmaceuticals in the river Foss. And you can also see that the concentrations we detect are a little bit higher. Now that perhaps, if you were listening when I was talking about the treatment works, might be a little bit surprising, because the river Foss, we actually have quite a small treatment works. It's 18,000 people. Whereas in the main river, we've got 200,000 people emitting. But this difference in the exposure, the fact we see higher concentrations, we think is primarily down to the differences in the flow of the two rivers. So the river Ouse is quite a high flow river. You've got lots of dilution. The river Foss, even though you've got less wastewater, treated wastewater going into it, is a much lower flow system. Therefore, you're seeing higher concentrations. And we can delve into this data. So we get monthly data, and we can begin to look at relationships between things like prescription. And what we find is that for quite a lot of molecules, we're able to sort of map on what the doctors are prescribing in the city with what we're detecting in the river. And the river is really giving us a feel for what's being prescribed by the GPs. So does that matter then? Well, scientists, I think, for the last 20 years have really started to think about the effects of pharmaceuticals on ecosystems. And the driver behind that really is the fact that these are biologically active molecules. So in us, they're designed to be taken into the body. They interact with a pathway or receptor, and they have the therapeutic effect. So that's how they work. The issue is that a lot of those receptors and pathways that the drugs interact with also occur in organisms in the environment. So a fish, for example, will contain almost all of the receptors that many of the pharmaceuticals we use today actually are targeted at. So if you've got these molecules going into the environment accumulated by a fish, then you might find, or you might expect to find, some type of effect. And there's a big literature now on effects of pharmaceuticals on organisms in the environment. What we've done is we've taken some of that literature and we've compared it with the data that we measure in York's rivers and also other English rivers we look at. And if we take ibuprofen, for example, which is a common over-the-counter drug that we use in the UK, what we find is that the levels of ibuprofen that we see in English rivers are pretty close to the levels where in the lab people have reported effects on fish hatching over very, very long exposure periods. If we take diclofenac, so diclofenac is an anti-inflammatory drug, and I'll talk a little bit more about that drug in a moment. The levels of diclofenac that we see in English rivers are quite close to levels where in the lab, groups have talked about effects on fish liver histology. So it does appear that certainly for some of the substances that we see in English rivers, the levels do seem to be high enough to potentially cause effect on aquatic organisms. We also think that they could be affecting terrestrial organisms. So we did some work a few years ago where we worked with one molecule. It was a molecule called fluoxetine. So fluoxetine is an antidepressant. The common name of the product it's used in is a product called Prozac. And what we did was we spiked worms with concentrations of fluoxetine that you'd expect to see in a wastewater treatment works, like the trickling filter I showed you earlier. We fed those worms to the birds, and then we looked at the effect on the bird behavior. And we found that when we did that exposure, the behavior of the treated birds was different from the behavior of the controlled birds. So the feeding behavior of the birds changed. And just the reason for that is if I went back to the slide where I showed you the trickling filter, when you go to these sites and you did go in with a spade, they're absolutely packed with worms. And you find that birds are living around the sites and they're basically using the trickling filter as their local food resource. So you do get that exposure pathway happening. And this is just one molecule. If you think that those birds will be actually exposed to a mixture of molecules, the effects could be even worse. And then the third concern that we've looked at in the UK rivers is the issue of antibiotics. And we find that the concentrations of some of the antibiotics that we see in UK streams are higher than those where in the lab effects on algae have been shown. So we expect perhaps effects on primary production in our rivers. But also what we're beginning to recognize is that these antibiotics in the rivers could be selecting for antimicrobial resistance in bacteria in the rivers. And if that resistance gets back into the animal population or the human population, that then could be contributing to the AMR crisis. And there was a recent paper I came out in, I think it was in January, where they'd actually quantified the global deaths from antimicrobial resistance back in 2019. And they estimated that it's 1.2 million extra deaths are caused by AMR in 2019. And we think that potentially environmental exposure is contributing to that number. And I'll come back to that a little later. So in the UK then, we're detecting pharmaceuticals in our rivers. The levels that we see in some cases are high enough where we expect effects to be occurring. And there is also this concern about human health effects through AMR. But what about more broadly? Well, this paper came out in 2016. This was a desk-based study. It was performed by a German group. It was led by Tim Alsterbeek. And Tim and his group worked for the German Environment Agency. He did a project for them. And what they did in that study was they brought together all of the monitoring data that had been reported in the literature from around the world to try and get an understanding of what we knew about pharmaceutical pollution globally. This figure or map basically sums up what they found. So the different countries in the map are colored based on what we know about water pollution from pharmaceuticals. So if you have a dark red country, such as the UK or the US, that means that there are data from somewhere between 100 to 200 different pharmaceuticals. Red is 31 to 100, but you can see that as we go down towards the yellow, for the yellow countries, there's only data for a handful, 1 to 3 different pharmaceuticals. And then if you look at the grey countries, these are countries that have no data at all. So you can see that for some reasons, regions of the world, particularly Latin America and Africa, we basically know very little about the levels of pharmaceutical pollution. And that then raises challenges in terms of assessing what the risks are. And that could be really important because we know from some of the data that have been produced over the years that these types of environments could have very, very high levels of pharmaceutical pollution. This is a photo from India. It was taken in a region called Hyderabad. It was taken by a Swedish group. And basically what they were doing is they were looking at the manufacturing areas in Hyderabad. So a lot of the manufacturing that occurred in the US, in Europe, I guess also Israel, has now been exported. So it's now done in China, in India. And what this means is you've got these big regions of pharmaceutical manufacturing. And in Hyderabad, what happens is the waste from that manufacturing, or at the time, the waste from that manufacturing was tankered to this facility. You can see the tankers emptying the waste into the facility. So it's into that tank there. Now, the problem is that that tank is connected directly to a tributary of the Ganges River. So effectively what you've got is direct emission of the pharmaceutical waste that contains antibiotics into a tributary of the Ganges River. And in that study, they did some analyses of the water in the system. And the biggest concentration they saw was for a molecule called ciprofloxacin. So ciprofloxacin is a fluoroquinolone antibiotic, and they measured concentrations of 31 milligrams per litre in the water. Now, to put that into perspective, if you and I went to the doctor and the doctor prescribed us ciprofloxacin, and we took that ciprofloxacin, and then I came along with a needle and I put it into you and I sucked out some of your blood, I analysed it for ciprofloxacin, the concentration in your blood would be about 3 milligrams per litre. So you can see that in the water, we've got a concentration 10 times greater than what would be in the blood of a patient being treated with ciprofloxacin. And that's really concerning in terms of AMR selection. And it's something that the industry are now aware of, and the bigger companies are now working to address the problem. If we go back to diclofenac, diclofenac also gives us reason to why we should be thinking globally. So back in the 1980s, there were certain areas of India and Pakistan that had very healthy populations of vultures. But over a period of about 20 years, what happened was the populations of the vultures just crashed. They went down and down and down. And this was a quote from someone from the Bombay Natural History Society. So what they said in the quote was, back in the 1980s, there were 4 million birds. So that's 9 different species of vultures, 4 million, many of them. But as we moved to the late 90s, that number had dropped to 60,000 birds. So a massive decline in the population of those birds. A lot of environmental forensics was done to try and work out what was causing the decline. And the ultimate conclusion was it was down to exposure to diclofenac. So what was happening is the people in the area, they were using diclofenac to treat lameness in their cattle. When cattle were dying, the carcasses were lying around, the vultures were coming in, they were gobbling up the carcass, getting a dose of diclofenac. And due to differences in the biochemistry of the vulture to the human, they were much more sensitive to the exposure than we would be. And it caused something called visceral gout. And over time, it effectively resulted in this massive decline in the population. And again, that's something that NGOs and others have now been working to correct. So diclofenac has now been banned for use in animals in those areas, and they've replaced it with a less toxic alternative. And they're also working to reintroduce and recover the vulture populations. So hopefully I've convinced you that actually taking a global perspective is really important. And we decided about 3 years ago that we would begin to address this issue of lack of data globally. And we established the Global Pharmaceutical Monitoring Project. What we did is we reached out to collaborators from around the globe. So it was over 100 different organizations that we collaborated with. We got Benny in the Zoom room. So Benny was one of the collaborators. And what we did was we sent out kit to the collaborators. This is a photo of the box that the collaborators received. In that box, there was vials, syringes, filters. You can see a little bucket there, and also freezer packs. And what happened was the collaborators would then go out to their local river and take samples along the local river. Because many of you are in Israel, I think, I thought I would take the Israeli example. So this is the system that we looked at in Israel. So this is the Nablus River and the Alexander River. So you can see that we've got a simple diagram of the river system. You can see that there's a wastewater treatment works on that system that we also sampled. But effectively what we did was we looked at different points along that system, right down to where the Alexander River emits into the Mediterranean Sea. And then we looked at the concentrations of 61 different pharmaceuticals in each of those samples. And in total, we sampled about 1,050 different sites across the globe. and that covered 104 different countries. And 33 of those countries had never been monitored before. So this is the first data ever on concentrations of pharmaceuticals. So here's just a few pictures. So this is actually the Alexander River. So you can see it's a very, very low flow system. And that explains why we see some of the results that we saw in that water. This was actually one of our MSc students that came over with me to work with Benny and colleagues. And I think she's wearing a Hebrew University Jerusalem hat. So that's keeping the sun off. So that's the Alexander River. This is sampling in Nairobi in Kenya. So this is Lydia Alaka, who was our collaborator in Nairobi. This is Lydia, I think, taking some samples from a small river that goes through the university campus in Nairobi. And then this is one of the European sites. This is in Tübingen in Germany. And that bridge is going across the Ammer River in Tübingen. And you can see the sample being processed. So that sample would then be frozen and then shipped back to us at York, where it'd be analysed for 61 different pharmaceuticals. Now, again, I could spend a lot of time talking you through the data because there's a lot of data. In total, I think we've got, because it was done in duplicate, we've got about 120,000 different data points. But I'm just going to give you a high-level overview of what we find. And the first thing I'm going to do is just give you what we found in terms of total pollution at the most contaminated sites. So what we have here is we have the average concentration for each sampling campaign. So a sampling campaign would be the Alexander River campaign, for example. And what we've done is we basically determined a total concentration for each site along that campaign, and we then averaged those total concentrations. So it gives you a broad level of the contamination. And you can see from this that firstly, the Alexander River does figure. So if you go to the bar 7th in from the right, that's where the Alexander River is. So there we see a sort of average total concentration of somewhere between 10 and 20 micrograms per litre. Some of those sites would obviously be higher than that. But the most polluted sites we see in Asia and also Latin America. So the most polluted site was Lahore in Pakistan, and the second most polluted site was La Paz in Bolivia. And you can see the photo there. That's actually one of the sites in La Paz. And again, you can see that's a very, very low flow system. And I think that particular site is actually adjacent to quite a big area of manufacturing. So we think that area is actually being contaminated by manufacturing waste. But generally what we find is the highest concentrations are in Asia, some of the Latin American countries, and also Africa. And then you get Europe appearing a little bit, and the only appearance of the US is Dallas, which is the second bar in from the right. And obviously we have a lot more of those data. We have this data for all 104 different countries and the whole range of campaigns. I think it's 138 campaigns in total. In terms of the concentrations of the individual compounds then, this is what we found. So this is just a box and whisker plot of the concentration ranges. So you've got the median, the upper and lower quartiles, and then the maxes and the mins. You can see that the highest concentrations that we see in terms of the maximum is paracetamol. Perhaps that's not surprising because that certainly in the UK, that's probably the biggest used pharmaceutical, although wastewater treatment is quite good at removing it. Caffeine is the second, and I'm sure that that's not from pharmaceutical use, but it's really a reflection on the fact that we all drink so much coffee and tea. But you can see that metformin, which was the drug that we found in the York system at highest concentrations, that's number 3. And in terms of the median concentration, it's actually the highest. And there were compounds that we didn't detect. So we didn't detect some of the penicillins. Miconazole is an antifungal compound. Norfluoxetine is a metabolite of fluoxetine. Oxytet is an antibiotic. and then sertraline is an antidepressant. So there were compounds that we never saw, and there were a few compounds that we only saw occasionally, which is shown on this slide. So this is the detection frequencies. So again, we have the different active ingredients on the x-axis. The detection frequency is a percent of the total sites that we monitored on the y-axis. And you can see that on the left there, we have another compound. This is carbamazepine. So carbamazepine we detect in over 60% of the locations that we monitored. And Benny, for example, has done quite a lot of work on carbamazepine uptake into crops and also human exposure. So that's quite an interesting molecule in terms of human exposure and crops. And also I think it's relevant in Israel. But again, you can see metformin, again, we're seeing at a similar detection frequency, about 60%. Now the colored bar chart basically represents the complexity of the mixtures that we see. So it's probably a little bit difficult to see, but on the x-axis, we have the number of compounds detected. So that ranges from a single compound right through to 34 compounds. So you can see there were quite a lot of sites, so 100 sites where we only detect one compound, and there were actually some sites where we didn't detect anything. So we did one really interesting campaign, which was in a village in sort of up in the sort of upper reaches of the Amazon River. And this was a village that basically had not been touched at all by Western people. So we actually managed to link up with someone who lived in this village, and they took the sampling to the village, took the samples and brought it back. And we didn't find anything or no known pharmaceutical in that water. But if you look at the graph, you can see that there are some samples where we're seeing 34 different pharmaceuticals in that water. So some of these locations have quite a complex mixture of pharmaceuticals. So what we're seeing from the global study then is that quite a large proportion of sites are contaminated with pharmaceuticals. In some of these sites, we're seeing much higher concentrations that we see in the UK. And that then raises the question, well, what are the effects? And what we've been doing is similar to what we've done in the UK. We've been using published data on different effects of pharmaceuticals. So we've been using apical ecotoxicity data. So this would be data that would be done by industry as part of their regulatory testing. So it'd be things like effects on algal growth, Daphnia reproduction and growth, Daphnia mortality, and also fish reproduction and growth. We've also used what we call non-apical ecotoxicity data. So these are sort of less standard endpoints. So this would include things like biochemical effects, effects on behavior. So these are things that regulators don't think about, but could potentially be very important. We've looked at resistance. So we've taken some of the safe values for resistance selection. I won't talk so much about that today. And the final approach we've used is we've used human to environment read across. And there what we've done is we've taken information on the therapeutic concentration in the human blood, in the plasma, and then we've used quite a simple model to back calculate what concentration in water would be required to meet that concentration in fish blood. So it's a sort of read across approach. It's a sort of early warning approach. And then what we've done with each of those different data sets is we've compared the concentrations that we see for each of the drugs with those different safe values to get an idea of whether we should be concerned or not. We've also, for the apical data, we've also not just thought about the single compounds, we've also thought about the mixture effects. And this, again, the idea here came out of some of the work that I did with Benny. So Benny and I, we hosted, in York, we hosted one of Benny's PhD students, Tom, and Tom did a really nice study of the Alexander River, and we began to look at risk in the Alexander River. So we built on some of the thinking that we did with Benny and Tom. But what we found from this analysis is that about 4 in 10 of the sites that we monitor have concentrations of at least one pharmaceutical of concern. And it's a mixture of things driving it. It's a mixture of the apical endpoints, the read across endpoint, but you can see that the non-apical endpoints are really figuring. And as I say, we don't really know what the environmental relevance of these are, but they could be very, very important. So there's a move now to begin to think about effects on fish behavior, for example, as part of the environmental risk assessment process. And when we look at the compounds driving those effects, then we find that there's about 23 pharmaceuticals actually responsible for those effects. And that includes things like antidepressants, stimulants, antibiotics, antihistamines, diabetes treatments, and painkillers. I think the other thing that we're finding is that at some of these sites or some of these samples, you would expect, based on this analysis, multiple effects on multiple endpoints. So what we've done here is we've developed these spider diagrams where we've got the different endpoints in the spider web, and we've taken the top 10 most polluted sites, and then we've mapped on the effects on those different endpoints. So you can see that at some of these sites, you've got concentrations affecting fish growth, fish biochemistry, Daphnia length, algal growth. At others, you've got affecting fish behavior, less of an effect on fish biochemistry. So we're beginning to sort of see multiple effects on multiple trophic levels in the system. And I think this is quite interesting moving forward. What we're interested in doing now is beginning to sort of take this type of approach and see what that means in terms of ecological functioning. So that's quite theoretical. So we've also been doing some experimental work. And this work has only really just kicked off quite recently. So we will be producing more and more on this as time goes by. But one of the things that we've done is, obviously, if we've got 1,000 locations across the world and we've got data on concentrations across those 1,000 locations, it's quite difficult to test the toxicity of all of those different samples. So what we've done is we've done a sort of a statistical analysis. So we've used clustering to group sites based on the pattern of pharmaceuticals that you see at the site. And we come up with about 7 different mixtures that represent the types of mixtures that you see in the environment across the globe. We've then taken those mixtures and we've selected a high concentration, a medium concentration, and a low concentration. And when we then tested the effect of those mixtures on organisms. And the organism that we started with is a really simple test. We worked with garden cress because it grows very, very quickly. So this is a plant. We grew it in silica sand silica sand soil, and the solutions were then added to the soil, and we then looked at the effect on the plant. And this graph here shows some of the data. So this is for biomass. So on the left there, you have the control, and then the different sets of bars are the different mixtures that we tested. You have the low, medium, and high concentration. And what we find is that almost all of those mixtures and all of those concentrations that we test do seem to affect the plant biomass. So if, for example, in a country people are using these sort of rivers to irrigate crops, it could actually be affecting crop productivity. And this is something that we want to look at in a little bit more detail. We've also done similar work with invertebrates, and we find that some of these mixtures just completely kill Daphnia. So we started running chronic studies with Daphnia, and we found that a chronic study will take about 21 days, and we found that after about 15 days, all of the Daphnia had died, so there wasn't even any reproduction. We didn't see that in all samples, but we saw it in some of these samples. And I think this highlights that we need to move away from working and thinking about single molecules to looking at the effects of mixtures. So what's driving all these impacts and effects then? Well, when the sampling teams went out to their sites, they would usually take notes and provide us with information on the characteristics of the sites. And this is just a word cloud where we've taken some of the most polluted sites and we pulled out the types of things the sampling teams talked about. And these most polluted sites, they're associated with, I guess, fairly things that you'd expect. So sewage is mentioned, waste is mentioned, hospitals are mentioned, farming, agricultural runoff, rubbish, industrial discharges. And also you can see it's spelled wrong there. There's a little word called exhauster, and I'll talk a little bit about that at a moment. There were some sites where there's mention of these things called exhausters. And when we begin to look at sort of some of the most polluted or highest contaminated sites, what we find is that it's areas where you've got a little bit of political instability. So the Alexander River is an example. Levels, we saw very high levels where the river crossed from Palestine into Israel. Population density played a role. So Hong Kong, we saw very, very high concentrations. And that's because you had a very high concentration of humans going into quite a slow flow system. Delhi would be another example. Quite a lot of sites had very poor wastewater connectivity or waste connectivity. And some of these systems were in a very dry climate. Again, the Alexander River would be an example of that, but Las Vegas, Madrid, Denver, and Tunis would be others. And I just want to show you a few photos of some of the more contaminated sites. So this was the site that was most contaminated with antibiotics. This was a site in Bangladesh. It's very close to a manufacturing site. So you can see some houses in the background. And behind those houses, there's a pharmaceutical manufacturing area. And in that water, we have found levels of metronidazole, which is an antibiotic, 300 times higher than the concentration that we think is safe for resistance selection. So it's possible that the sediment and the organisms in the sediment in that area could have quite high levels of resistance genes. This is another picture of Nairobi. So this is a less pleasant picture of Nairobi to the one that I showed earlier. You can see at the top there, you've basically got a pipe with lots of solid material coming out of it. And that pipe is basically sort of moving untreated sewage and wastewater from the local population into the river. So that is completely untreated. And this is another photo of Nairobi. This again, the river itself looks a little bit more attractive, but this was actually one of the most polluted sites. And what we did was we went to Google Street View because the sampling team, Lydia and her team, talked about the evidence that there would be exhauster trucks around the area. And we just went a little bit up from the bridge that this photo was taken at with Google Street View, and this is what we found. And these are exhauster trucks. These trucks basically go around Nairobi and emptying out the septic tanks of people's houses and hotels and businesses. And what we think is happening, and Lydia suggests that this is the most likely cause, is these trucks are then coming to this park here. They're emptying their contents directly into the river. And that's why we see very high concentrations of pharmaceuticals and antibiotics in particular. And again, at that site, we find concentrations of antibiotics well above the safe levels for resistance. Moving to Ghana, this is a site in Accra in Ghana. This is actually a dump site. It's a dump site that is quite famous. It's been in the UK newspapers. And it's one of these big super dumps where waste, particularly e-waste from Europe and North America, is shipped to and disposed of. And you can see in the background there, you've got a river and an informal settlement behind the river. Now, the informal settlement will probably be contributing pharmaceuticals, but we think the dump site is also contributing pharmaceuticals. So it's probably not just e-waste that is being disposed of at this site, but also medical waste. And there is some evidence that I've seen suggesting illegal export of medical waste from developed countries to some of these low or middle income countries, such as sort of Ghana and this site in Accra. So this could explain that situation. But then there's also just general littering. So this is going back to Nairobi. This is a photo of one of the stream banks in Nairobi. And I think you can probably see there's in the front there, there's definitely a tube of toothpaste being disposed of. And we suspect, you could well have just old pharmaceuticals, waste pharmaceuticals, old over-the-counter medicines in that mix, and that then is leaching into the river system. So I think to begin to sum up then, we've known for a long time that pharmaceuticals are released to the environment, And we know that they're released at different stages in their life cycles. So they're released during manufacturing, during their use, and also during the disposal. But until now, we've tended to focus on countries like European countries, North American countries. Increasingly, we've had data for China and India, but for lots of areas of the world, we've simply had no data at all. And what the global pharmaceutical studies allowed us to do then is basically perform the most extensive monitoring study of pharmaceuticals ever. I think it's probably the most extensive of a micropollutant in rivers ever. And we've included 33 countries never studied before. When we take that data, what we find is that actually the areas that we've been focusing on up until now tend to be the cleaner areas. And the lower middle income countries that we've ignored are the ones that are most polluted. I mean, that's probably not surprising, but we've now got the numbers to back that up. And what we think is driving it is a whole mix of things. It's manufacturing, it's poor waste and wastewater management infrastructure, and it's also possibly, for antibiotics, for example, overuse and over availability of some of these molecules. We find painkillers, anti-inflammatories, antimicrobials, diabetes treatments, anticonvulsants, antacids, antihistamines, and beta blockers at the highest concentrations. And when we take those concentrations, what we find is it's a fairly significant proportion of the sites that we monitor that have levels where we might expect effects. But I think what this is showing, and this is what we're going to be exploiting going forward, is that if we can begin to combine some of the more rapid new analytical methods that we have available with data from the literature and a better connectivity of global science. So I think we're all much better connected despite COVID. So we are able now to work together to ship things easily. I think we can really begin to build on this work and understand what the scale of pollution is globally of rivers and other systems. And this is what we're going to be working on moving forwards. So just to finish then, much of what I've talked about is published in 2 papers. So the monitoring study itself was published in PNAS back in February. You can see the author list there. So it was a massive list and a massive collaboration, that study. And then last week, a follow-up paper, which was the ecotoxicological effects assessment, came out in environmental toxicology and chemistry. So if people are interested in that, I'd be quite happy to send copies. I can send the PDFs to Orly, for example, and she can circulate them. The study itself has received massive press coverage. So I think it's really helped to raise the profile of pharmaceuticals and pharmaceutical pollution around different stakeholder groups. So I think what we're working towards is make the decision makers much more aware of this issue, and hopefully that will result in action and actually mean that things begin to clean up and get better. And we do have a website. So if you want to tap into the data, all of the data are available on our website. So go to www.globalfarms.org. On that website, there's lots of photos of the different sampling locations. You can meet the team and the collaborators. You can hook into the publications. And as I say, you can download and access the data. And I think there's an interactive map that you can play with to look at the concentrations in areas that you're particularly interested in. So at that point, I will stop. I think I've just gone over 45 minutes. So I'd like to thank you all for your attention and for coming along today. I hope it was interesting. And if people have any questions, I'd be more than happy to answer them. So thank you so much.
Dear Alistair, thank you so much. That was fascinating. I really enjoyed your lecture, and it also worried me very much. Yes, we have some time for questions, about 10 minutes. So if anyone wishes to ask, please raise your hand and unmute yourself. Yes, I see Adi Salomon.
Yes, thank you very much for your fascinating lecture. And as it was mentioned before, it's wow, like I don't know what we can do about it. So I'm a professor in the chemistry department in Bar-Ilan University, and we use some, we detect some pesticides, herbicides, also pharmaceutical residue in water. But I just wanted to ask you whether there are some countries that are regulate, or what, which are the countries where you have regulation, like probably in Europe it's more regulated than others, or like if you compare, for example, Paris to other dense country places, whether you can say something about it. It's also interesting.
Yeah. So I think pharmaceuticals themselves in Europe are not directly regulated at the moment in rivers. So the European Commission are thinking about it. So they have a program where they have what's called under the Water Framework Directive. So that's the directive that does all the water quality monitoring. They have what's called the watch list. The watch list is a list of chemicals that they think may be of a concern that in the future they might want to regulate, but they're not regulated at the moment. So there's no quality standards at the moment. And there are a few pharmaceuticals on that watch list. What we think might happen is some of those pharmaceuticals might actually then become priority substances and quality standards would be set. But at the moment, they're not regulated directly. I think perhaps what works in the favor of some of the European rivers is that obviously other emissions from wastewater are regulated. So things like some of the pesticides, some of the nutrients, some of the metals. So I think because of that, the level of wastewater treatment legally has to be, and the way it's monitored and regulated is so tight that wastewater treatment processes in Europe are much better than they say are in Africa. And I think that will have, even though it's not directly aimed at pharmaceuticals, that will have a positive effect on some of the pharmaceuticals. So paracetamol, for example, we find, you know, when we go into UK rivers and we look for paracetamol, even though we use, I think it's something like 660 tons per year of paracetamol in the UK, even though, and that's by far the biggest used drug, you don't detect much of it in the river, and that's because 99.9% of it is removed by the wastewater treatment processes that we use. Whereas if you go to Nairobi, because you don't have that wastewater treatment process, that's where you'd start detecting paracetamol. Now, just as an aside there, we do, and this is actually quite, it's probably not an issue in Israel, but in the UK, we're beginning to realize that the water companies, when the wastewater treatment works, when we have a lot of rain, which I know you do occasionally, but not as much as we do, when we have a lot of rain, the wastewater treatment works designed to receive a certain volume of water. And if we have too much rain, what happens is what we call combined sewer overflows kick in. And what happens is the wastewater diverts around the works directly into the river. When that happens, we then start seeing paracetamol because you haven't got the treatment. And what we're finding in the UK is actually these combined sewer overflows are flowing much more regularly than they should do. So some of them are just flowing for hundreds of hours each day. So that's something, as I say, that in the UK, they're getting quite worried about.
Thank you.
I think there's a question from Ronit.
Yes.
Yeah. Thanks. I was wondering, in Israel, we use a lot of recycling water. And there have been done some studies in Israel that shows also there's a lot of leftovers medication. So I was wondering if what's the best way to detect it, like around hospitals, around other places. And we have the recycling water given to trees and to vegetables gardens, and then we get it back through the food. So it's a huge problem also in Israel. And what's the best way to eliminate the medications,
Yeah,
the drugs?
I think it's quite system specific. So I think where you've got good wastewater treatment infrastructure, one of the simplest ways is to just add a tertiary treatment, something like granular activated carbon or ozonation. So that in the UK that would work. That would remove a lot of the compounds, and I guess it would probably work in Israel as well. Benny might want to sort of comment on this. The challenge I think is going to be where in systems where you don't have any wastewater infrastructure at the moment, like those photos of Nairobi. And there, I think we're going to need a different type of solution. So a lot of the emissions that we were seeing in Nairobi are, from informal settlements. There's no piped sewage system. They're just pit latrines that are overflowing into the river. You saw the waste disposal. So I think the waste disposal possibly could be dealt with, so that you could possibly introduce improved waste collection. But the actual wastewater itself, I think possibly what they need is sort of some of these more nature-based type methods that are low energy, can be applied diffusely, so you can put them almost on the household, but they need to be very cheap. I think that might be the solution, rather than coming in with a big costly engineering solution, which I don't think would work. In terms of the hospitals, you mentioned hospitals. There's some really, again, I think this would only work in countries like the UK and maybe Israel. There's a really interesting initiative in the Netherlands where a hospital in Delft has introduced what's called the pharma filter. And basically what this is it's an integrated waste and wastewater management system for the whole hospital. So this is a new hospital, and they've built the waste and wastewater management system especially for the hospital. And they've designed it to recover plastics, recover the metals, prevent any microbes getting out, and also minimize the pharmaceuticals coming out of the hospital. So they have a small treatment plant in the hospital that has ozonation to remove the drugs. And I think that's really interesting. And they're trying to sell that concept to other new hospitals. But again, I think it's probably only appropriate for economies that have that can afford it. I don't think it would work in some of these other countries. I think we need cheaper solutions that are low energy.
Thank you.
I think Nahama has got her hand up.
Yes, Alistair, we are a little over an hour. If it's okay with you, then we can have the next 2 questions. So,
okay.
Okay, thank you.
I have a question. Are the levels of pharmaceuticals so high that if you drank water based on which is coming from these rivers, it would have an effect on people, not other than animals? I'm talking about.
Yeah, so we have done a little bit of work on that with the data for York. So we've calculated acceptable daily intakes based on what we see in the rivers, and then we calculated acceptable daily intakes for the humans, and then we compared that to what we find in the rivers, and the levels are quite a lot lower than ADIs. So in terms of risk to human health from consumption of water, particularly in the UK at least. Now, whether it's going to be true in some of these more polluted areas, but in the UK at least, I don't think it's a high priority. I'm not saying it's not an issue, but I don't think it's as high a priority as some of the ecological effects and the antimicrobial resistance. Okay. But yeah, if you calculate the ADIs for some of these things, you'd need to drink water maybe for tens of years, 2 liters a day to get to that concentration. Okay. But then I've had discussions with people who said, but still, we can't rule out the fact that we as humans will be exposed from the day we're born to the day we die to these very, very low concentrations. And to actually assess the effect of that's very difficult. So I'm not ruling out there being a risk, but I think in terms of priority, I don't have that as a top priority at the moment. I think the plant exposure will be greater, and that's something that Benny's working on. I think that the exposure in situations where you've got wastewater reuse, the exposure from consumption of plants is probably greater than drinking of water, but a lot of the focus has actually been on drinking water risk.
Thank you.
Yes, now for our final question by Uri, go ahead.
Oh, thank you. I couldn't find the hands up. Can you say a few words about the circulation time of a few of the compounds that you've looked at? Because some of them may be in the system for ages, some of them have been maybe an incident, sporadic incident that's happened in the past. You could still see it, but then 6 weeks later, maybe it's gone.
Yeah. For these molecules, so we do have data on persistence of some of these molecules, and the reason that we don't detect some of the molecules that I talked about, so the penicillins, for example, is exactly that. They're hydrolytically unstable, so they don't hang around very long. And there are others where they will persist for a very long time. So many years ago we did quite a lot of work on the tetracyclines, and the degradation rates of tetracyclines are very, very low. So if you're going to the terrestrial example, tetracyclines will stick to sludge. They're also used in animals. If they go onto soil, we did studies showing that effectively a year later, when you add the next lot of slurry or sludge, there's still tetracyclines in the soil. So the persistence varies, but one of the issues is that certainly in the river systems in the UK, because we as society are effectively using these things continuously, the persistence that comes out of the equation, because we're so populated, you've got towns emitting continuously these things into the environment, that they're actually, even if they're degraded fairly quickly, they're still there all the time, just due to the fact that we're using them all the time. But if you stopped using them, then they would go down, some of these.
Thank you.
Good. So first of all, thank you to Professor Boxall for being here. I think it was fascinating, and you could see the participation and the questions. You made a lot of impact, I think, today. And I will conclude by inviting you to our next webinar on July 19th by Semyon Polinov. The webinar will be about the spatial and temporal assessment of oil spills in the Mediterranean Sea. We will send you the flyer with the link for the webinar in the next few days. Stay tuned. And this webinar will also be available on our YouTube channel. You will also receive a link. And thank you all. And have a nice rest of the day. So, bye.
Thank you.
Thanks,
Bye-bye.
everyone. Bye.
Thank you.